don't like westerns.
Yeah, I like sci-fi much better, or horror films.
John Wayne is so weird.
Oh my god, Faith, my teacher said I have this thing DNA in me. I'm gonna die.
Oh no, silly Nilly. DNA is in everyone. It stores genetic information that affects the way we look.
That's devil talk, Faith. Shame.
Tie her down.
No. We're gonna explain DNA to you, okay, Colleen?
Okay.
How do you want to explain DNA to her?
Let's rap to her. DNA is good in every way.
No, Gabby. Faith, show her this.
This is a double helix.
No, Faithykins, that's a double. Felix.
Oh, right. This is a double helix. It is made up of two nucleotide chains connected by weak hydrogen bonds. A nucleotide consists of a sugar called deoxyribose, a phosphate group, and a nitrogenous base. These are connected by covalent bonds.
Stop, it's my turn. I want to talk about how it's replicated, my favorite subject. First, helicase separates the two nucleotide chains, like helicopters chopping through things. Then DNA polymerase creates a complementary strand for each nucleotide chain. This creates two identical strands of DNA, each with one old half and one new. This is called semi-conservative replication. This all happens in the nucleus. La 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 la.
The devil's possessing me because of this.
Get the chains.
Yes!
There are 4 different nitrogenous bases that can be present in DNA. They are adenine, guanine, cytosine, and thymine. A and T go together, and C and G go together. Whoa, Gabrielle, what'd you do to Colleen?
Nothing.
Okay, fine, I get it now.
That's right. Yeah!
Okay, so RNA. I know that it has a sugar ribose and a phosphate group and a pinch of heaven. I mean, a nitrogenous base. But how is it different than DNA?
Well, it's single-stranded and contains a different sugar, and it also replaces the nitrogenous base thymine with uracil.
Get off my woman!
Like urine?
No.
No, little Kipper, let Emma continue.
As she was saying before Urine Girl over there interrupted, adenine pairs with uracil, A and U.
They're golden.
Like me.
So A cheats on T with U? That's like a primetime soap opera. Go on.
Well, there are 3 types of RNA. The first is mRNA, which brings information on how to make proteins from the nucleus to the ribosomes. Then there's transfer RNA, which brings amino acids to the ribosomes to build proteins. And finally, there's ribosomal RNA, which makes up the ribosomes. Go on, Emma.
I will. I'm going to tell you more about RNA. The process that transfers the information from DNA to RNA is transcription.
Oh, I know this. Okay. Once the DNA strands have separated, transcription begins. It starts in the promoter region, and the RNA polymerase produces a strand of RNA that is complementary to the strand of DNA.
Did you read my diary? It's my dream to have somebody tell me about RNA. But then, I started killing everybody with bowling balls, and when I woke up, I smelled bowling alley.
Okay.
Now, Poppet, you have to remember that in transcription, thymine is replaced with uracil.
Oh, and I have to pee.
Transcription continues until an enzyme reaches the termination signal. The DNA and RNA are released, and RNA goes into the cytoplasm.
Wait, what happens next?
Translation, the process of making proteins from mRNA, baby boo.
mRNA travels from the nucleus to the ribosomes. Then tRNA brings the amino acids to the ribosomes, which begins pairing up with their corresponding codons.
Codon love.
It begins at a start codon, or AUG. mRNA will continue to read each codon and send tRNA to get the right amino acids until a stop codon is reached. These include UAA, UGA, and UAG. This makes a protein.
So basically, DNA is transcribed into mRNA, which is then translated into an amino acid chain that makes a protein.
Exactly.
Are we done yet?
All we need is
really sure. It could be due to a mutation.
She's an alien?
No, well, maybe. What's a mutation?
A mutation is a change in the DNA of an organism.
One example would be a deletion. This is when a DNA sequence loses one or more of its nucleotides.
Like this.
And insertion is when an extra nucleotide is added to a DNA sequence.
And a substitution is when one nucleotide is replaced by another nucleotide in the DNA sequence.
If a nucleotide is added or deleted, it incorrectly groups codons. This could be a big problem, because then the mutated DNA codes for the wrong amino acid, affecting the protein.
UV radiation and chemicals can cause mutations.
High energy radiation can cause free radicals. This can cause breakages in the cell's DNA.
The cell cannot fix this deletion.
Chemicals can cause insertions, deletions, and substitutions. These can cause modifications in the base structures.
For example, nitrite, a drug, substitutes cytosine for uracil in a DNA sequence.
Well, is there any technology that can help?
Yeah?
Oh my god, you're so stupid! Gosh!
DNA technology is used in many different ways. DNA fingerprints are composed of specific fragments of an individual's DNA.
These DNA fingerprints can be compared to those found in samples of blood or tissue found at a crime scene. This helps us to identify the culprits of many different crimes. But I haven't gotten caught yet.
DNA technology can also be used to produce prescription medicines and vaccines. Bacteria can grow in large vats to produce protein, which many medicines are made from. Insulin, the drug used to treat diabetes, is made from this DNA technology.
Farmers can also use DNA technology to become more efficient. They can genetically engineer crops to become resistant to pests and diseases. This makes farming less expensive.
So what's DNA again?
Oh my
my goodness.
You've got to be kidding me.
Away, panda, away!